Thank you so much, and sorry for keeping you for the lunch. But they just insisted to make a food safety presentation before lunch, so that's the organizer take. So thanks for the conference team for having me. I am from the United Arab Emirates University, from the College of Agriculture and Veterinary Medicine. This is a technical talk or a technical session, so excuse me for the title, a little bit complicated for some. But our research is focusing about a very important One Health food safety issue, which is Campylobacter. Campylobacter is the most important, most frequent foodborne pathogen. It's a bacteria. Mainly when we are talking about Campylobacteriosis in human, we are talking about two major species, Campylobacter jejuni and coli, and they are the most common bacterial cause of human gastroenteritis worldwide. Why this is important for the topic of the conference, One Health? Of course, it's a bacteria running at the human-animal food interface. So it is across the road, and when you are translating this exposure to clinical or health outcome, it is responsible for more than 95 million cases annually worldwide. So it is a very important foodborne pathogen. And what is worrying us, and I think we heard about this topic a few times throughout this morning, the increasing burden of antimicrobial resistance in this bacteria, where some of the empirical famous antibiotics like fluoroquinolone and the macrolides are also increasingly resistant in this foodborne pathogen. So it's really growing to be a global control challenge. Chicken meat or poultry chain is quite blamed as the most frequent food source for transmission of the infection with Campylobacter, the foodborne exposure. It is running from the farm, where biosecurity at the farm will be enabling these bacteria to be introduced. It is not harming the chicken. It is living with them very friendly. The best temperature to grow it is in the lab is 42 degrees, and the body temperature of the chicken is 42 degrees. So this is like a best mix and match. And when slaughtering is done under some imperfect condition, cross-contamination is happening, and it is a surface contaminant. So it is basically found on the outside of the chicken, like the skin. This is moving to also the consumer when where unhygienic handling or relatively speaking also eating raw or grossly contaminated chicken in domestic kitchen and also in restaurant could be leading to cross-contamination, and this is really the main source of human exposure. Relating to the United Arab Emirates, poultry meat is by far the most consumed animal protein. So we are eating a lot of chicken. Some estimation is about 55 kilograms per capita, which is putting us among really the major chicken eaters countries worldwide. But unfortunately, to a certain extent, there is little data, about baseline data about the prevalence of this foodborne pathogen, which is an important challenge for food safety. So, as you heard this morning, the UAE is a country where there is a lot of innovation, a lot of commitment to evidence-based food safety policy. And it is not really strange where universities like United Arab Emirates universities meet with Abu Dhabi Agriculture and Food Safety Authority to do collaborative research. And we started this project, which has been just concluded a few months ago. The focus was about the two major foodborne pathogens, actually Salmonella and the Campylobacter. And we were interested to get an idea about what is the baseline status of this pathogen at the farm level, at the abattoir, and ending up also by characterizing them at phenotypic and also genotypic level. So it is a kind of farm to fork first baseline in the Emirate of Abu Dhabi. You hear this morning also that the majority of the agriculture sector and also the poultry industry sector are located around the Emirate of Abu Dhabi. So it is not really strange that this collaboration comes between United Arab Emirates University in Al Ain and ADAFSA, the Abu Dhabi Agriculture and Food Safety Authority. So we done a very comprehensive tracing across the poultry industry from the slaughter batch, from the farm level throughout the processing. And for example, per one slaughter batch, meaning group of chicken going from one flock house, slaughtered in one day in the abattoir, and then going to the consumer, to the supermarkets. So intensively, we sampled, for example, from one slaughter batch about 30 samples, 15 CCAM content. So this is telling us what is inside the gut. So this is giving us also indication about what is the situation in the farm itself where the chickens are originating. And also it was very important to understand the dynamic of the contamination throughout the processing, throughout the key processing steps like defeathering, evaporation, and then the final washing and cooling. And basically this is a chicken which is sent to the supermarket at end of the day. And for each integrated producer, which is a trend here in the United Arab Emirates, so major companies owning their own farms and also their own slaughterhouses and their own distribution chain. And we sampled about 10 slaughter batches from each company. We'll just give an example about one of the results per one of these companies. So over 10 slaughter batches, we sampled 300 samples throughout the processing, as you can see here. And we used state-of-art technologies and the standard methods and also multi-TOF techniques, which is a high throughput mix between laser technology and spectroscopy to identify the bacteria. And also in our labs, we have set up also to do whole genome sequencing to understand also better these bacteria to the genome level, and also to get much better information about its risk characterization and the pathogenicity potential to the consumer. So for example, in this company G, we sampled, as you see here, 10 slaughter batches. All of them are positive for Campylobacter, and the prevalence after each of the abattoir processing step is quite high. And with also identification at the species level, we could find a predominance of Campylobacter coli over Campylobacter jejuni. This is a little bit surprising for us because the trend in many countries is a little bit the opposite. Anyway, what you can see here, the industry is receiving the chicken, which is highly colonized with high load of Campylobacter inside their intestine. So as you can see here, the load is above 7 log colony forming unit per gram. So this is really highly concentrated load of Campylobacter inside their gut, and they don't show any ill sign. The industry is doing a great job. As you can see here, they try their best to reduce the load throughout the processing, after defeathering, after evaporation, till after chilling, but it is still a challenge. As you can see, after the final step in the abattoir, we still have a lot of carcasses going to the supermarket with a bacterial load which is more than 1,000 colony forming unit per gram, which is quite an issue for Campylobacter because it is just a few hundred bacteria are enough to make someone sick. So this is really quite a challenge, and as you see, yeah, you can say garbage in, garbage out. Unfortunately, if you are starting at the farm with highly colonized bacteria in the gut of the chicken, the industry is doing its best, but it cannot really survive that much. This kind of violin chart is also telling the same Yes, the industry is capable of doing reduction, but unfortunately, it is, as you can see here, reduction and stabilization. So it is not the expected more and the more and the more reduction. The load in the gut of the chicken is very high, and this is why the industry or the industrial abattoir line, however it is efficient, modernized, similar to any European setup of an abattoir, but still also find it very challenging to keep reducing more. As you can see here, the reduction is not really very much going in a proportional way. It is a reduction and a stabilization, which is really highlighting again the whole key. The earlier that you are controlling it at the farm level, the better that you are giving the industry or the abattoir processing line a better chance to defend or to try to control it to a much more efficient way. Genomic is a fancy tool used in a lot of research, but here we are using it actually in a more applied way. So we are using here or try to put a proof of concept that certainly these expensive tools of genomics can be telling us also a lot about what is happening regarding cross-contamination and biosecurity. Just a couple of examples here. In this green area, you see in this tree, some groups looks very similar, but actually they are coming from two different slaughter batches originating from separate farms. So they are different slaughter batches from different farms, and they are still sharing the same genotype. So that means certainly there is a cross contamination. And the same also for these clusters where they share the same sequence type, so they are the same family more or less, but actually they are coming from the same farm, different slaughterhouses, which is telling us cross-contamination is happening at the farm, carried over at the abattoir, and this lead to persistence of some able to persist very well genotype. So this is a great example telling us that whole genome sequencing and genomic tools is not just a research tool, but it can be purpose used to identify biosecurity shortfalls and also cross-contamination. Genomics or whole genome sequencing also could add a lot of value to build or to understand what we call genome informed risk characterization. So we know that it is Campylobacter, but how risky or how problematic it to human or to food safety from a food safety point of view. You can get really deep knowledge about all the gene portfolios, and I'm not going really to go into details, but just to tell you, even two species in the same chicken, they carry a lot of differences between the gene and the genetic makeup regarding to antimicrobial resistance. And the same also to inform about risk characterization, virulence factors throughout this pathogen is very important to map. As you can see here, this is a heat map, which is telling us that there is big variation between the genotypes as well. So certain essential genes responsible to illness or pathogenicity not there in some species are more expressed or more presented in some species. So whole genome sequencing is really a very useful tool, not only for characterizing the bacteria, but also to trace the genomic traits which could be impacting health, food safety, and also illness consequences out of this if there is a foodborne exposure to this pathogen. So to conclude, this initiative, this project between United Arab Emirates University and ADAFSA is really adding value to food safety, public health, and putting a great example for One Health case at the poultry sector. It helps a lot in addressing much of the gaps about some of the most important foodborne pathogen in our most popular animal protein consumed here in the United Arab Emirates. The project point to the multifactorial nature of the contamination at the slaughter houses. It also elaborate about the usefulness of genomic-based tool or highly sophisticated genome-level studies to also provide some very sophisticated evidence about cross-contamination at the abattoir, clear emphasis about the importance of hygienic practices throughout the processing, and also highlighting that the intervention as early as possible at the farm level will be helping the production and the processing at the factory. I just give an example about company G, but actually in our project we extended this extensive sampling frame to 4 integrated poultry producers. So we sampled more than 500 samples from 39 abattoir visits, from 30 farms, and from about 70 flocks, broiler flocks, throughout Abu Dhabi Emirates. So hopefully this is all will be data delivered to ADAFSA, providing them with science-based evidence for improving and also adding value to the poultry industry in the country. So I'm trying my best to be on time. Thank you so much, and thanks to my team and to ADAFSA and for their support. Thank you so much.